what this gene card is? My genome, whole genome map is given in there. Just like your credit card, you swipe this card and you can see my genome and you can predict based on this, after 10 years, what will be my health, what possible diseases I can suffer. Or now what they are moving into, they will not call you for the interview, they will ask you for your gene card. You swipe and it will tell you we want to have you or we don't want to have you. Love affairs will be broken. You take the gene card of the girl, your bride and bridegroom, and you swipe them and you say these are the possible combinations. Disease or non-disease. You say no, I don't like my son or daughter to have curly hair. So she say no, I want. Finish. Thank you, I don't want to marry you. Yani this is, we are entering into a very dangerous zone where the whole social structure is at stake. You know, when they were doing this human genome project at that time, I remember I was in Singapore at that time, and there was a Sri Lankan professor. His name was Arif Bongso. Arif Bongso was working on embryonic stem cells. And somebody, he was presenting in a conference, and I was sitting like you are sitting here, and he was saying, somebody asked him, Professor Bongso, the government of Singapore doesn't allow embryonic stem cell research. He said, I will leave Singapore. He said, where will you go? I said, any country which will allow me to do this embryonic stem cell. They said, no country in the world is giving permission to do this. Wallahi, I still remember, he's still alive. He said, I will go in the international waters. I will carry my lab in a ship and I will work over it. But I will not give up my research. This research was stopped because the humans, they started doing something which is gene cloning. So if you have one bush, and if you have 100 bush, we are trying to get rid of one bush as president of USA. But if you clone 100 like this, the next century will be gone like this. So humans will suffer. So it was him who stopped the research. He said there is no human human clone. Anyway, Dr. Your diseases can be classified as single gene disorders. We call them as, that is, one gene is malfunction. We call this as monogenetic. Oligogenetic, when you have more than one, less than 10. Polygenetic, when you have multiple genes which are involved in it. There is also a possibility, inshallah, when you go in 3.1, I will deliver your lecture on, you know, you have bacteria, you have viruses, you have also something called environmental pathogens. Stress is an environmental pathogen. Just like bacteria can infect you, stress can also infect yourself. So at that time, inshallah, I'll be talking to yourself how the environmental factors interfere and interact with your genes to give rise to certain disease process. So there are some diseases which are traumatic in nature. And when these two combine together, that is the environmental factors and the genetic factors combine together, we say these diseases are multifactorial. There are some genetic factors, there are some environmental factors. Environmental factors interacting with your genes and giving rise to diseases which are called multifactorial. Diabetes is a multifactorial disease. Heart failure, blood pressure, they are multifactorial. No one gene you cannot point this gene is the one which is responsible for this. Okay? So monogenetic diseases, many of them you have already come across, like SCID, severe combined immune deficiency, in which you lose your B lymphocytes and T lymphocytes. SCID, hypercholesterolemia, cystic fibrosis, sickle cell anemia, hemophilia, they are all monogenetic disorders. Just one gene. You see how precise is Allah subhanahu wa ta'ala's creation. One phenylalanine is lost in the whole gene of cystic fibrosis. One phenylalanine is modified in its place, and your chloride channels in the lungs will stop working. understand me. This is the level of PCL. I'm talking about the whole 100 billion cells. And in the 100 billion cells, one phenylalanine is misplaced. And you lose your ability to breathe. Is it? Then you have the polygenetic disorders, which are heart diseases, cancer, diabetes, alcoholism, schizophrenia. All these are polygenetic disorders.
Doctor, can we consider type 1 diabetes as a monogenetic
No.
disorder?
Because nowadays it's considered to be more immune related. The immune systems start destroying this. So it's not monogenetic disorder. Now the nucleic acid as drug molecule, the modern day concept of pharmacological intervention is intended to introduce a transgene. Transgene means a gene which is not part of my body but has been transferred into there. So gene therapy or transgene therapy is the same thing. Do you follow?
Yes.
If I ask you, a gene has been inserted in there, or I say a transgene has been included into, so it means the same. So transgene means a gene that has been transferred into the genome from outside or into the nucleus. We call this as a transgene. Intended to introduce a transgene into the target cell. Target cell means the cell which I want to insert this gene in there. Favorably modify the expression of the gene of interest to prevent, stop, or revert a pathological process. Is it clear?
Yes.
This is called gene therapy. Doctor, until now, from 1989 to 2018, more than 2,500 clinical trials in the gene therapy they have been carried out in different diseases. And if you look at the breakdown, astonishing here, only US is contributing 65% of these clinical trials and 23% from Europe. That makes it about 88%. And the remaining whole world is contributing 12%. And out of this one, these gene therapies are mostly about some antigen or some cytokines or some receptor modifications in the biological system. And if you look at the types of the diseases, the most focused type of the disease for gene therapy is cancer. And if you look at the vector means a vehicle, inshallah I'll be talking to you in more detail. A vector means a vehicle which is used to transfer a gene into a cell. We call this as a vector. And if you look at the most commonly used are the adenoviral vectors, followed by retroviral vectors, followed by naked DNA. Transfer of DNA without the help of any other assistance. So these are the three major forms of transfer of transgene, that is the use of adenovirus, the use of retrovirus, and the use of naked DNA. They are the three most prevalent forms of introduction by which the methods by which the gene is introduced into the cells. Is this clear, doctors? Do you have any comments or anything to say?
No.
Doctor, the first ever gene therapy drug that was marketed was way back in something 2004, when this Chinese company came up with a product which is called Gendicine. The first product which was ever launched in the market came from a Chinese company, and that product was called Gendicine, which is a recombinant adenoviral vector carrying p53 gene. Now p53 is related with tumor. So this was for the treatment of cancers. So what they will do was, they will insert, they considered that the cells in the tumor, they have mutated p53. Let us insert a transgene of p53 which is normal functioning. Can we cure these tumor cells by giving them normal p53? So what they did was, they developed an adenoviral vector carrying a p53, normal p53 gene, and they did intratumoral injection. Where the tumor was, they injected this gene in there. So directly transferring this gene into the tumor cells, and they ultimately realized that putting this normal p53 gene into the cancer cells, it helped the cells stop their proliferation, and the tumor started shrinking, and also became more responsive to radiotherapy. So they were very happy when this product was launched. But then this was followed by another company, which was Introgen Therapeutics. They came up with a similar product, which is called Advaxin. Advaxin and Gendicin, they were meant for the same purpose. The only difference was here the promoter used in the adenoviral vector was slightly different. It was a cytomegaloviral promoter. That was the only difference between the two products. Otherwise, they were meant for the same purpose, and the strategy used was almost the same. So I repeat it one more time. The first ever gene therapy product was Advaxin, Gendicin, which was a recombinant adenoviral vector carrying p53 gene for intratumoral injections. And the second product was Advaxin, same strategy, same thing. The only difference was that the promoter in the vector was different. You know, every gene has a promoter.
Yes.
So they made use of cytomegaloviral vector promoter rather than anything else. Doctor, $1 million product, the Luxturna. Luxturna, $1 million for the treatment of inherited blindness. One injection of this into the eye is because retinal dystrophy was caused by malfunctioning of RPE65 gene. So they made use of a normal gene and directly injecting into the eye, retina of the eye. And they say that this is going to help a blind person start seeing the things. But the cost, $1 million. $1 million and you can see the colors of the world. This Dutch, they came up with a product which is called Glybera. It's a life-saving drug. These patients, they have this disease because of LPDL, lipoprotein lipase deficiency. Because of the lack of this enzyme, the fatty metabolism, that was interrupted. And it causes the fat deposition in the blood vessels to the level that the blood vessels become totally occluded. And that can lead to the death of the person. So they came up with this product, which is called Glybera, with this normal gene insertion, costing 1.1 million euros for one injection. And then, doctor, the latest in this series is called Zolgensma. Now, Zolgensma is for the little babies in which there is a gene which is called SMN1, survival motor neuron 1. That causes, the malfunctioning of this one gene causes spinal muscular dystrophy. And because of this spinal muscular dystrophy, the babies cannot sit upright. They are all the time lying down. So what they do is they make the injections and insert the normal gene in there. Now, doctor, whether this is Advaxin, or this is Gendicin, or this is Luxturna, or this is Zolgensma, or Glybera, what we are doing in there, we are taking a normal gene and inserting them into, and injecting them directly into the human body. This method of gene therapy we call as in vivo gene therapy. In vivo. There are three things. In silico. What is in silico? In silico? Huh? No.
In labs?
In the lab. On the computer. Silicon Valley. In silico, on the computer. In vitro,
I know them.
in the lab.
In the lab?
In the lab.
Okay.
In vivo,
Human.
directly in the biological system. Ex vivo,
Outside.
Tissue
Outside.
from the outside.
ex
Which one is that?
in vivo is an animal.
We said no.
No, in vivo is the human, and it's not only human. Anything which is done in the biological system is in vivo. Vivo means live. Huh? Vivo means live. So in vivo, there again I repeat it, 4. In vitro,
Cube. Cube.
in the lab,
Yeah.
in silico, on the computer, in vivo, inside the
Human, but-
biological system, whether this is mouse, rat, human, pig, dog, whatever, whenever you will penetrate the defenses of the biological system, this is in vivo. Ex vivo,
Halving cell than the lab.
very good. You take the biological sample, you take the cells of your interest out of the biological system, take them in the lab, genetically manipulate and put them back. This is ex vivo. Is this clear?
Great.
So in silico is not a gene therapy. This is just your computer modeling. Right? So you have in vivo, in vitro, and ex vivo. All these 4 procedures, if I ask you gendicin, delivery of gendicin or P53 using adenoviral vector by intratumoral injection is an in vivo process. Use of Dexterna to deliver RPE65 gene into the retina of the eye, in vivo gene therapy. Is this clear, doctor?
Yes.
Clear.
Similarly, this Glybera, direct injection into the patient, in vivo therapy. So all these examples which I have talked to you until now, they all belong to in vivo gene therapy process, and if you are asked at the exam, please note it down. Yes, please.
Usually these drugs, the patient needs just one.
Sometimes they need only one injection, sometimes you may have repeated cycle.
It will
It
be-
depends, you know what, it depends on what type of the vector have you used. I will come to this in the end. What type of the vector are you using? Say for example, it will be too early for people. I have to tell them what is the vector, then I will tell them how they were. Okay? But just remember, the number of times you need the injection depends on what type of the vector and what type of the product you are looking for. Okay? I'll come to this inshallah.
And all gene therapy is in vivo.
That I also come back. That is in vivo and ex vivo is also done. You see here, that's why I put in here, all these products are in vivo gene therapy products. There is an ex vivo gene therapy procedure. Here you have the patient, you take his cells, genetically modify, and then put them back in. You can make use of autologous cells. Autologous means coming from the same person. If I'm the patient, you take my cells and use them and manipulate them and put them back in my body, autologous. Allogenic, you take Jeru cells, genetically modify them and put them in my body. This is allogenic.
Same species.
Same species but a different donor. And if I take pig cells or dog cells and put them in my body, this is xenogenic. Do you understand what I say? Yani the other day they did a pig heart in a human. Do you remember? Pig heart in a human. What type of the transplantation is this? This is xenogenic transplantation. My heart transplanting to someone else, allogenic transplantation. Is this clear, doctor?
Yes.
Very good. No, doctor, this is something which we have already done in our discussion, the chimeric T cell receptor cell. This is an ex vivo gene therapy procedure. A patient, we take his T lymphocytes, we genetically modify this, then culture them, increase them in number, and put them back into the patient. This is called ex vivo gene therapy. If I ask you, CAR T cell is what type of the chimeric T cell receptor antigen receptor is what type of the gene therapy? This is ex vivo gene therapy because you take the T lymphocytes of the patient, culture them in vitro, genetically modify them, then expand them to increase the number, and then put them back into the same patient. This is ex vivo gene therapy, but this is autologous ex vivo gene therapy, taken from the same person, putting them back into the same person. Yes.
Sorry, Doctor, we don't speak about that in the tutorial. Can you briefly, what's the...
Yeah. Now, CAR T cell is even ahead of. I've just published one systematic review, sending out a new concept that your drugs can be classified as pharmaceuticals, ordinary chemicals, biopharmaceuticals, biomolecules like antibodies, and they can be living biologicals, living biodrugs, that they are literally living drugs like cells. CAR T receptor is a living biodrug. A dead T lymphocyte will not give you any effects. The T lymphocyte which you are injecting back into the patient must be alive. Stem cell therapy, the cells you are going to put in the body of the patient as a drug must be alive, not dead. So this is a new class of drug in pharmacology emerging, which we call as living biodrugs. Drugs which are living cells that can be used for the treatment. Now, CAR T cell, chimeric antigen receptor T, this is a big technology, but to summarize in one word, one sentence, T cell receptors, T cells, they are presented an antigen before they can do something. CAR T cells are the T lymphocytes which have been genetically modified, that their T cell receptor has been tagged with another antibody on top of this. Now this antibody can recognize the target cell. It doesn't need T and T cell antigen presentation to the T cells. They can self-recognize the antigen, go and bind with this, get activated automatically, and destroy the target. We call this as chimeric antigen T cell receptors. This is one of the most and the latest drugs which are coming in the market, and there are already two marketed products. One is Chimera and one is called Yescarta, and they recognize CD19 on the tumor cells, and they can destroy the tumor cells without antigen being presented to them.
Basically, they take T lymphocyte and present to them the tumor receptor.
Say it again, please.
You take the T lymphocyte and present to them the tumor receptor.
No. What you do in there is that you have, if this is my T cell, which I have taken from the patient, this is the T cell receptor.
Needs B cells, but now it doesn't.
No, it doesn't need. It needs APCs, antigen presenting cell. Now, if this is the T lymphocyte, this is the T cell receptor on top of this. But generally what happens, this is the antigen presenting cell. It processes the antigen with MHC2 and bring it to here to make the recognition. The chimeric T cell receptor doesn't work like this. This is a T cell with T cell receptor, and on top of this, by genetic engineering, I put an antibody with this T cell receptor. This antibody can directly recognize the antigen. Now, here is a tumor cell. This antibody will go and bind with this. When they bind together, it sends the stimulus to the T cell receptor. This T cell receptor will send the signal downstream to activate the T lymphocytes. Now, these T lymphocytes, once activated, they start proliferating, they become active, they start attacking the antigens. And is this clear?
Yes.
So basically, we put the T lymphocyte with antibody to.
Yes. So we genetically engineer the T lymphocyte T cell receptor with an antibody.
We insert the gene in the T lymphocyte, or
Yes, we do it by this way. We genetically engineer the T lymphocytes to start expressing this antibody on the surface.
Okay.
Anything else?
So what does chimeric mean?
Chimera is a cell preparation which is available in the market. Do you understand? So Chimera and Yescarta are the T cell receptors the genetically modified T cell receptors against CD19. Is it? Very good. So this is the historic perspective of gene therapy, how it has developed and how different types of the products like Libera and how over the time period they have been marketed. So Zolgensma is the latest one which has come in the market in 2019. If it is clear, doctor, I go further. Now gene therapy has 4 different types. One is called gene supplementation, as the name is indicating. Gene supplementation, gene suppression, gene replacement, and gene repair. They are the 4 different approaches by which the gene therapy can work. Supplementation is a genetic mutation in there, and you supplement this with a normal gene. This abnormal gene, mutated gene is still there, but I supplement this with a normal gene. So this will start producing the enzyme or protein or cytokine or growth factor of my interest. Is this clear, doctor? This is gene supplementation. Gene suppression, this is a mutated gene which is giving rise to certain enzyme or something, which is creating a problem. I suppress this. Put an inhibitor which can suppress this gene. We call this as gene suppression therapy. Gene supplementation, you supplement the abnormal with the normal one. Gene suppression, that the abnormal gene's expression is suppressed. Replacement, you cut the abnormal gene out and you insert the normal gene in its place. This is gene replacement therapy. You replace the things. And gene repair therapy, there is one mutation in there. You remove it and repair this. This is gene repair therapy. Any one amino acid or any one nucleotide is missing in there or abnormal, you only replace this part and the remaining gene is the same. This is gene repair therapy. Is this clear, doctor? Who will tell me what are the 4 types? Who will revise for me what are the, yes please?
There is gene supplementation where we supplement the defective gene with another
Very
gene.
good.
Gene suppression where we suppress the mutated gene to suppress their
Expression.
expression. We have replacement where we replace the defective gene or nucleotide.
Very good.
And we have gene repair where they repair the amino acid.
Yeah, where we gene nucleotide or the amino acids is repaired inside, and that is called the gene. So 2S and 2R.
What's
SS.
the difference between supplementation and repair?
Repair is,
To change the amino acid.
yes, repair is that this part is damaged, I take this and repair this one.
Supplementation.
Supplementation, that I insert the whole part, new gene in there.
Both will be exist here in the gene.
Say it again.
Both will be existing in the gene.
In both of the cases, when I'm doing replacement, I say, for example, in the whole genome, this part is missed, is the abnormal. I replace the whole chunk of this. This is gene replacement. I remove the abnormal gene and replace it with a new one. But repair is, just like your tire is there, have a mismar, and you do the repair and finish, the tire remains the same. But there is replacement that you go for the whole replacement of the tire. You understand me?
And supplementation.
Supplementation, that the abnormal gene is still there, but you put the new one in there.
Thank you.
For the
Like
therapy.
still, okay.
Yes. Is this clear, doctors?
So the suppression will be separate with normal or just separate?
In supplementation, the normal gene will be dominant. The normal gene will dominate the functioning of the mutated gene.
So this normal you will insert.
It will insert the normal gene in there. No, doctor, all these procedures can be done in the somatic cells or in the germ cells. If it is done in the germ cells, we call this as germ cell therapy or germline cell therapy. And if it is done in the somatic cells, we call this as somatic cell gene therapy. Is this clear, doctor? No, what do you think has the advantages with the germline gene therapy, and what is the benefit of somatic cell gene therapy?
Germline, I think the new gene will replicate all over the body.
No. Germline gene therapy, doctor, is for the next generation. Somatic cell gene therapy is for my son. So there are moral and ethical issues involved in there. I want my babies to have white complexion and curly hair or blonde type. I ask, go for germline gene therapy to change the gene expressing the color of the hair in the sex cells. Now the moral therapy is, what if my son doesn't like it or my daughter doesn't like it? Can you control his wishes? So this is the moral and ethical aspect of this.
But if we're talking about disease,
Pardon?
if we're talking about disease,
That is different thing. But I'm telling you, what are the possible consequences that can occur? The morals and ethical issues which are involved in it. If it is for the treatment of a disease, yes, very good. But if it is for the sake of cosmetics and aesthetics, then there is a moral issue with this. Somatic cell gene therapy only restricted to the person receiving it. Is this clear, doctors? The somatic cells are modified for this, like intratumoral. It's a somatic cell, you inject directly into this, so the gene therapy effect is not going to the next generation. It will remain with the person. This chimeric T cell receptor is a somatic cell gene therapy. It doesn't go to the next generation. Is this clear, doctor? So 4 types: supplementation, suppression, replacement and repair. All 4 can be repeated for germline gene therapy and somatic cell gene therapy. Germline for the next generation, somatic for the person receiving it. Very good? Now doctor, how you compare gene therapy with protein therapy? There is another approach which is protein therapy, that I am missing interleukin 1 and you inject interleukin 1 in my body. This protein, you have genetically engineered IL-1, so you give it to me. Or these dialysis patients, they are receiving EPO, erythropoietin. This is protein therapy. Every time they do the dialysis, they are giving an injection into that dialysis fluid, and this EPO goes into the biological system, let the kidneys make more this bone marrow. The bone marrow to make more bone cells. Now, the biggest difference between the gene therapy and protein therapy is, what do you think?
that's
Protein
temporary.
Temporary.
protein therapy will give you
Protein therapy, you have to do repeated kind of. It's normal like any drug treatment. Gene therapy is one-off treatment. It's curative. That is symptomatic. Protein therapy is symptomatic treatment. The gene therapy is a one-off curative in nature. You replace the normal gene and the person cures forever. Other than that, here you make use of some viral vectors or some vectors to transfer the gene. This is without any vector treatment or anything. No, doctor, ethical issues, as I said, good and bad aspects. I already told you, germline gene therapy has the moral, that stigma onto this. Other than that, one thing which you might have noticed, Luxturna 1.5 million, Zolgensma 2.5 million. Is this only for the rich?
Yes.
You know? For the time being, yes it is, but in the long run I hope inshallah it will be for everyone. Doctor, what is the gene therapy procedure? What it takes you to do the gene therapy? This is one of my project. I did with these fellows. He was a Russian, he was a Chinese, Vietnamese, he was Indian. All of us, we got together and we made a project to artificially create or experimentally create myocardial infarction in mouse and rats. And we said, okay, how we did it? We ligated the coronary vessel and created an infarct in there, right? Then we want to create new blood vessels around this to bypass this blood vessel which we ligated. And then, if you look at, we created all these new blood vessels in the region. We took the heart out, did the sectioning, and that did the staining, and we find a lot of new blood vessels bypassing the vessel which we have ligated. We call this as biological bypass surgery. Normal bypass is done, you take a vessel and ligate it over. No need. We can grow new vessels in the surrounding and bypass the one which is blocked. This procedure is called biological bypass surgery. Now this was an experiment which we created in the rats to do this biological bypassing to create new vessels in the infarcted region so as to restore the blood and the muscle will become healed. How we do it? We identified the affected gene, we cloned the gene to have more of it, and then we loaded this gene into a vector. And then these vectors we targeted onto the cells where we want to grow the vessels. And then we made use of a delivery strategy, and ultimately the genetic material gets integrated into the. Do you understand this? So it all starts with what type of the gene do you need, and then you clone this gene, then load it into a vector, transfer this vector to the target cell, gene is inserted in there, and then it expresses itself to give rise to. Is this clear up to this? What we did in this experiment was we created, we identified that we can make use of VEGF, vascular endothelial growth factor, which can create new vessels. IGF to make the cells proliferate, HGF and SGF to enhance their mobilization and their survival. Now putting all of these 4 genes into one viral vector, we created this, and then we rather went one step further. We put this vector into the cells, stem cells, and then transplanted them into the heart to create and achieve this result. Is this clear, doctor? This is the fundamental, the basic procedure of a gene therapy, that you identify the gene, you clone it into, and then you insert this into a vector, and then you transfer it to the cells of your interest. Very good. Doctor, the first ever gene therapy trials, the first ever, was done in 1990. There was a little girl, her name was Ashanti De Silva. She was born with a defect in an enzyme, a defect in a gene which was encoding for an enzyme called adenosine deaminase. Adenosine deaminase. Now what this enzyme do, this is what it does. Very small thing. Will lie in the canvas of my whole body. This is so tiny little dot. You see, here is adenosine. Here is the amino group. You don't need to remember these formulas, just to make you understand how small things can make how big difference. Presence of this amino group here, NH2 group here on the adenosine, was loss of it. This enzyme causes removal of this amino acid to convert adenosine into inosine. Now, accumulation of adenosine is very toxic for the cells. So this, as part of the Purine metabolism, what has to be done, that this adenosine has to be all the time converted into inosine, which is innocuous, non-toxic. And in the whole process, only this amino acid needs to be removed by the presence of this enzyme. This baby was missing this enzyme, and the enzyme was non-functional. So she was not able, her body was not able to convert adenosine into inosine. And this inosine was getting accumulated in T lymphocytes and B lymphocytes. And they are very sensitive to the concentration of adenosine, and they were dying. Because of this, she was suffering from lymphopenia, and her blood, the T lymphocyte, B lymphocyte count was less than 200.
Check.
So here you see this doctor, French Anderson, he said, I have a cure. Let us take her T lymphocytes and put the new gene in there, and then proliferate these cells, increase in number, and inject them back into the patient. So he did this, and now you tell me what type of the gene therapy is this?
Living brother.
Ex vivo.
They took the T lymphocytes, genetically modified for ADA, and put them back into the patient. And she survived, and now she's, mashallah, young girl, running around. So this is a typical example of an ex vivo gene therapy. This is what they did. Here's the patient, take the T lymphocyte, genetically modify these with the normal gene, proliferate these cells in vitro, and then inject them back in the patient. This is the process by which ex vivo gene therapy is carried out. Am I clear, doctors? Doctor, this skin is also called bubble boy disease. Inshallah, when you go third year, fourth year, you will come to know that this skin, severe combined immunodeficiency, is also called bubble boy disease. Why? Huh?
Bubble.
Bubble boy. Now, why this is called bubble boy? This little boy was born, David, his name was David, was born in September 1972 in Children's Hospital in Houston. A few minutes after his birth, the doctors realized he had a problem. So they put him in a plastic bubble. You see here, this was his bubble. He grew up in this bubble for 11 years. Never came out. And here you see when he was like 5 or 6 years of age, NASA created a space suit for him. Because they said this baby is going to die inside the bubble. Let us bring him out. So they created a space suit for him. You can see him here wearing this space suit and playing with his sister. And now the doctors were waiting that his sister will grow up, become a bone marrow donor, and they will do the transplantation. And they were trying to keep him like this forever. Here you can see he's talking to his son, to his doctor. Here you can see he's talking to his mother. And his mother is saying, oh, you are the star of the moment, and all this kind of stuff. And you read this one, very interesting. Now, at the age of 11, at the age of 13, when his sister was grown up enough to donate the bone marrow, she donated, they did the transplantation, and because of infection, he died. So after him, they named this kid as bubble boy disease. So it's the story behind this, why it's called bubble boy, is because this David was kept in the bubble for almost 11 years of his life. He spent in there. Doctor, this is another, this is the one which I was talking about, this Chelsea Gilsinger, the first gene therapy death which occurred. This boy, this is his last word, he said, I die, it is for the babies in future. So he was suffering from a disease in which he was lacking OTC, ornithine carbamylase. This enzyme helps the conversion of ornithine into citrulline, and this is part of the urea cycle. And because of the malfunctioning of this enzyme, this boy was given bottles and bottles of OTC just to keep him alive. And I was reading an article about this, that he has hundreds and thousands of these bottles piled up in his room because he was using day in, day out for them. When he died after the gene therapy, the story which I told you earlier, Then his friends got together, his schoolmates, so they burnt his dead body, and when his dead body, these ashes were taken and filled in these bottles. And then they took it to a cliff where he used to go for his summer vacations and all that. And then they stood over there, then they sprinkled the ashes in the air in his memory. Well, this is how they celebrated his death. But anyway, I at that time when he died, I remember I was in the US at the time, and there was so much of gloom everywhere. And next day there were headlines in the newspaper, no clinical trials and all this kind of. Everyone was very sad because of his death and because of the stoppage of all the clinical trials which are taking place. Remember this husband and wife, a very sweet couple. I met them in Washington. They were working on creating a gene therapy for the colorblind people, those who cannot see the colors of the word. One injection, rods and cones will start working very nice, and the person can see the color. So,
Is it effective?
pardon?
Is it effective?
Yes, it is. So they, at that time they were working. I hope they have been able to complete their project. This we have already discussed in our class, and this is T-cell receptor. And she was, this little baby, Lala Richard, she was the first person to receive this CAR T-cell treatment. Cystic fibrosis, as I was telling you already, there is a gene which is called CFTR, which is the transmembrane reductance regulator. It is regulating the functioning of chloride channels. This one gene and one amino acid, phenylalanine at a position of 508, is making the chloride channels malfunction. And because of this, the chloride ions cannot be pumped out. When the chloride ions sit in there, they hold the water as well, and they hold the sodium as well. So because of this, the secretion of these cells, they become sticky in nature. So that is preventing the breathing and all this kind of the stuff, cystic fibrosis. Now in here, they have come up with a gene therapy for this cystic fibrosis. One is liposome based, in which the gene GL678 is delivered using a non-viral vector. I'll come to this a little earlier for yourself. So this is a liposome based and this is a virus based gene therapy. So what they do is they ask the patient to inhale this, and it directly goes into the lungs and it's taken up by the lung cells, and the CFTR gene becomes normal in there. Now, what type of the gene therapy is this?
In vivo.
In vivo gene therapy. Very good. And this is vector-based, viral vector-based, and this is non-viral vector-based. But in both of the cases, it is inhaled, it directly goes into the lungs, deliver the gene to the normal cells, to the disease cells in the lung, and that rectifies the genetic issue in there. So this is an example of in vivo gene therapy. Route of administration is not intravenous, it's not intraarterial or it is
Inhaled.
nasal. Nasal. These are other gene therapy trials. If you want to read more on this, you can always go to these. Last part of my topic, doctor. Vectors. What are different types of the vectors? And how they differ from each other? What is their efficiency? And how good they are in doing the. But we classify these vectors. Vector means a delivery system. The DNA uptake by the cell is very poor. Very, very little. There may be 0.1% or something like can be taken up by the cells as such. Unless and until you manipulate the cells to something. We classify these vectors or the carriers of the gene as viral vectors and non-viral vectors. Okay? Viral vectors, any virus you take and you modify this, not directly. Like I was talking to my people in pharmacology that there will be a time when COVID will be used as a vector to deliver the genes. We have already overpowered COVID, and I'm 100% sure because of its specificity and tropism for the respiratory system, if they want to do gene therapy for the respiratory system, I think COVID is one of the best examples. So, but they have to modify this vector, take the pathogenicity genes out, and insert the normal genes in there. So the vector will become non-pathogenic, but will be a carrier of your gene of interest. But very soon you will be hearing this thing. The ideal vector should be safe for use in the humans. Immune responsiveness should be less. Production should be easy. The capacity to carry the transgene should be large, because some genes are very large. They cannot be inserted in small vectors. So your vector should have enough capacity to carry a large gene. Must be targeted to the cell of interest. It must not be undergoing mutations afterwards. It should be stable, and it can be regulatable. Regulatable means once you have injected, then you have no control on this. You know, can we regulate this? Yani, for example, currently they have been able to create beta cells for diabetic patients' treatment. They take their cells, body cells, develop, induce pluripotent stem cells out of them, and then they convert them, these cells, into beta cells for insulin production. But the success is not there, because my beta cells respond to the condition of glycemic conditions. If the sugar level goes up, they start secreting. The sugar level goes down, they stop producing. So they are regulatable. They are sensitive to glucose concentration.
The feedback.
You know, but the new cells, the beta cells which they are generating from these iPS and stem cells, they are non-responsive to the changes in glycogen. Otherwise, they have already been able to have the cure. They can replace the beta cells. They have been able to produce cells which can very easily secrete insulin, but they are not regulatable. They just keep producing.
They don't respond
You know?
to that.
So that is leading to hypoglycemia and all that. So these are the characteristics of an ideal vector. The most important one, they should be safe for use in the human, no immune responsiveness should be there. And what are the four most critical steps that determine the success of any gene therapy process? This vector must bind with the target cell. Number two, it must deliver the gene of interest to the nucleus. Number three, the delivered gene must show transcriptional activity. And number four, that the product must be released from the cell into the surrounding. So these are the four critical steps which determine the success of any gene therapy process. Number one,
But not released as an antigen.
it should, that should be,
Express.
it should not be immunogenic. It should be acceptable. That is why ex vivo gene therapy is much better. For in vitro gene therapy, it's because your own body cells are producing something. So they will be producing something which will be acceptable to the immune system. Yes, please.
Should not be immunogenic?
Should not be.
What do you mean? Why should not be?
If the gene product, a protein, is immunogenic in nature, your body will give response, will destroy it very easily.
Yes, it's okay.
You know, so it should not be immunogenic. The product should not be. It should be acceptable by the biological system. So these are the. If someone asks you what are the four critical requirements for a successful of a gene therapy procedure, identification of the target cell by the and insertion of the gene into the nucleus, because if it is delivered here in the cytoplasm, it will be destroyed. So vector must take it into the cell's nucleus. When entering into the cell, this gene must be functional, so it should have a transcriptional activity. Otherwise, insertion of the gene is, and then the transcriptional product must not be immunogenic. It should be acceptable and released from the cell into the system. As I said, doctors, these vectors can be viral vectors, and and they can be non-viral vectors. Viral vectors, as the name is indicating, we have modified the viruses, different viruses which are available in the nature, and we have modified them and used them as carriers of our genes. These can be integrating viruses and non-integrating viruses. Sheikh Saab, this is for yourself. Integrating viruses are the ones which will take the gene and integrate into the genome of the cell, like your retrovirus, if you remember. If what it does, it takes the gene and integrates into the genome of the cell. We call them as integrating viruses. And non-integrating adenoviral vectors. They just deliver the gene. They don't care whether this goes into the genome or it remains episomal in nature. Episomal means not into the genome of the cell, rather as a plasmid. It stays in there. Extra genetic material. Extra chromosomal genetic material. So if someone asks you, if I give you retrovirus, lentivirus, adenovirus, adeno-associated herpes simplex virus, which one of these ones are integrating? Lenti and adenoviral vectors, they are the
Retro.
lenti and retroviral vectors, they are the integrating viruses. Whereas adeno-associated herpes, they are the non-integrating. Now what is the difference in the gene therapy response in these? When it is integrating is a long term, because as long as the genome is integrated, it will continue to express this, and the effect of the gene therapy will be long lasting.
If the cell divide will be part of that.
Every time the cell will divide, this integrated genome will be also divided until the telomere finishes, or some accident happens to the cell and it loses it. I remember I used retroviral vectors to deliver gene to pig hearts. I could I can identify the gene product after 1 year. So integrating are long-term therapy.
But to understand the non-integrating, the genome doesn't enter the DNA.
The non-integrating doesn't enter into the gene. That's why I say it remains episomal.
So it stays in the cytoplasm?
No, it remains in the nucleus, but not integrated in there. It remains as a plasmid.
So how does it replicate?
Every time the nucleus replicates, this also replicates. The plasmids also replicate. You know, but the fear why the non-integrating are short term, because there is a possibility that the plasmid is lost. But something which is integrated with the genome will not be lost. So the non-integrating, that is why these viruses, they have short-term activity. These have long-term activity. If you want a gene therapy effect to long-lasting, very good. Say for example, I want to grow new vessels in the heart for my biological bypass surgery. If I go for the integrating virus, what will happen? Even if the blood vessel has been made, it will continue to express VEGF and may end up having hematoma in there. So I don't need it. So we go for non-integrating virus. But I need insulin gene to be permanently inserted into the cell so that it can express insulin. I need something which can give me long-lasting effects for this. Is this clear, doctors? Now the non-viral gene, the vectors, doctors, you have gene gun, needle injections, electroporations. That what you do is you put the cells in an electrical bath, give electrical shock to the cells that increase the porosity of the membrane and help the cells to take up the DNA. But these are gene gun cannot insert a gene in a cell. This is for in vivo gene therapy. You have a gene gun and just inject this in there. On the other hand, we also have magnetofection, that you insert the gene using magnetic particle. You attach the DNA with some magnetic particle and use magnetic field around, and you make the they also use this for, I think, don't go longer. But on the other side, you have polymer-based gene therapy, which is liposomes, polyethylene, dendrimers, chitosan, nanoparticles. They are all polymer-based delivery vectors, but they are all classified as non-viral gene vectors. Viral ones, the ones which make use of viruses, and then you classify them as integrating and non-integrating. And non-viral vectors, you have some physical methods like electroporation, photoporation, sonoporation, magnetoporation, as the name is indicating. Sono means sound waves, magnetic means magnetic waves. Polymer-based, the most important one out of all this are the liposomes. Is this clear, doctor? Do you have anything in your mind that is unclear? Here, doctor, you have, see how these vectors choice has been done in the clinical trials. Adenoviral vectors are the most used ones, followed by retroviruses and naked plasmids, as I've already shown you. These days, people are liking to work with lentiviral vectors because of their superior quality. So why to make use of viral vectors? Because they're very efficient. They have tropism for certain cells, so they can very easily send the gene in there. They can identify their specific cells. They are very good for gene replacement therapy, and they are very easy to produce and cheaper, and can be produced in large amounts with very small money. But this slide needs a little bit of attention. How we modify any virus for use as a vector. You see, say for example, here is a wild type adenovirus. Wild type means the one which is found in nature and is pathogenic. If you look at the genetic composition of this, there are some genes which are determining the tropism of the virus. There are some genes which are for the structure of the virus. There are some genes which are pathogenicity determining, some immunogenicity, some for the resistance. What we do when we are trying to convert this wild type into a vector, we keep the tropism so that it can go to the cell of my choice. I replace some of these genes. I eliminate completely these ones so that it becomes non-pathogenic. I replace some of them to reduce their immunogenicity. And then persistence, I can keep them or improve so that the viral vector can stay for longer time in the. Now when I eliminate this one and I replace some of these genes, I create a space in there. And that space is used to insert the transgene.
The what?
Insert the gene of my interest, the transgene. Is this clear, doctor?
Yes.
So this is how we convert a virus into something which can carry my gene of interest. Now, someone might be thinking that I have removed the replication of the genes of the virus, the viral gene. How then I can multiply this virus to increase in number to inject? They're very intelligent people, doctor. What they do, they have developed certain complementary cell lines in the tumors. These cell lines have the same E1 which is needed by the viral vector to replicate. So what we do, we culture these complementary these plasmid containing cancer cells, cell lines, put the virus in there. The virus infects these cells, and when it infects, it goes and take the gene of replication from this tumor cell. When it goes into the infected tumor cells, it takes this gene which is needed to replicate. Okay? So it makes use of this cell's genes to replicate, keeps replicating in there and keep eating the tumor cells. When the titer of the virus is good enough, I pour off the virus from this petri dish to a container which doesn't contain any tumor cells, or it will stop growing. Do you understand?
So you mean that the complementary cells within the tumor cells, or they bring?
No,
it
we
back?
develop these cell lines, specific
And
cell
the-
lines.
And the doctors what?
Yes, and we put this E1 in there to make use of these special cells,
Correct.
tumor cells, to grow over E negative viruses. So when I put the viruses in there, they in fact use the E1 of the cell to replicate. When I take the virus out of the tumor cells, it again stops replicating.
Because there is no one.
There is no E1 available.
As if you're giving the tumor food and then you remove
Yes.
the food.
Is a tumor is a E1 provider to these virus cells. So when they have replicated, I just pour off this solution from the cells and finish.
And there is no tumor.
There is
Has finished the tumor.
is this yes. Shruti.
I didn't get this.
So here is very simple now. This is something which you can read very easily. Here is an adenovirus vector. It goes into the cell, goes into the nucleus, retain episomal. And these are the characteristics of the adenoviral vector. They carry the instead of reading all this, these are the advantages. They can be produced in high titer. They can in fact both dividing and non-dividing cells, wide tissue tropism, and can very easily be genetically modified. The only disadvantage is being able only to produce, to send the DNA as episomal. The expression is only going to be transient in nature. Okay? The second one is the retroviral vector, the integrating virus, because they will insert the gene into the genome. They will be long lasting. But here is the table which summarizes all this one, that retroviral vectors, they can only infect the dividing cells, whereas the adenoviral vector, they can do it both dividing and non-dividing. They are very broad in nature. So here is a direct head-to-head comparison of retrovirus, lentivirus, adenovirus, and adeno-associated virus. Any questions for this? They are the non-viral vector. Yes.
I didn't understand the part.
What? This one?
No, the briefing.
This one? The previous?
A1.
This one?
Yes.
This one. Who will tell Sheikhs of this? Sheda, you have to pay 5 riyal, whosoever will explain you.
I can explain.
Please, ask Sheikhs afterwards 5 riyals.
We have a virus. We want to replicate the virus with a specific gene that we prepared. So we put it in a dish with the tumor cells with E1. So the virus can come and attach to the tumor cells. And with the virus attached to the tumor cell, it begin replicating until it reaches to a certain point. Then we took the virus and put it in another dish where there is no tumor cells, so the virus will stop replicating. I think it will be ready for the
Yes. Very good. Sheda, is it clear?
Yes.
Or otherwise you can come and I will explain it to yourself. No problem. I'm just rushing because Dr. Majid has a lecture here. Done. Non-viral vectors, their biggest advantage is that they are cheap, easy to make in very large quantities. But the biggest disadvantage with the non-viral vector is poor transfection efficiency. Transfection efficiency, how efficiently it can transfer the gene. Viral vectors are good because they are highly efficient in delivery of the transgene. Non-viral vector, these liposomes and polyethylene and nanoparticles, their transfection efficiency is very low. Yani if adenovirus can infect 99 cells out of 100, the efficiency of liposome is, if you get 10%, you are very little. 1%, 2%, 5%. And these days some people claim 18, 20%, but I don't believe. Maximum which at least I have been able to achieve is 10%, not more than that. So that remains one of the biggest problem with the non-viral vectors. This is something which you don't need to remember. It's just for you as a filler. That when you are making the liposomes, what type of the different lipids can be used in there. So this is just for your general knowledge purpose, not for the sake of your exam. This is a head-to-head comparison between the efficiency of non-viral vectors with viral vectors. Gene transfer efficiency, if here is 3, it's only 1. And the cytotoxicity, almost the same. Immune response, viral vectors have more, here they have very less. They can do it dividing, non-dividing, they also can do it for dividing and non-dividing. Their capacity is limited, their capacity is unlimited.
For retrovirus, it should be divided.
Pardon?
Retrovirus should be divided.
Should be divided. Is it clear, doctor? And if in terms of toxicity you see, transfer of naked DNA, this is the minimum toxicity you can see. As you go up the ladder, your efficiency becomes poor, but the toxicity also becomes high. Is it clear, doctors? Very nice. Doctor, these are the last 3 slides. Very interesting. Well, like this is the heart, and I love this, the thinking process of the research. There's a process called biofection. Bio means biological. Transfection, use of biomolecules to do the transfection. Or instead of taking the viruses, which can be immunogenic and disease-causing, and problem of reversal, and then culture of tumor cells and all that, forget about this. Make a virus-like particle, genetically engineered. In the lab, you make use of different capsid monomers, put them together, make an artificial virus. And this artificial virus then can carry your gene without any fear, without any culture of the tumor cells. This process is called biofection, gene delivery vehicles using non-viral biological agents. One is virus-like particle. The second one, this is even more interesting. Take the RBCs. Now what is an RBC? RBC is a bag of protein with membrane, and inside is what? Hemoglobin. Remove the hemoglobin out of this. You can create a space. Okay? Now once you remove the hemoglobin, remaining is only a membrane bag. Load your gene in there. Now this is acceptable for the patient because autologous RBCs, I have removed the hemoglobin and I have inserted my gene in there. Now this is immunogenically more acceptable to my biological system. Very good. So this is called use of erythrocyte ghost. Erythrocyte ghost is like a gin. This is something which is not original. So what you do is you extrude out the hemoglobin, make this erythrocyte ghost, and then load with the gene of interest and put them back into the patient. Right? So your immunogenic problems will be gone. And the last one, use of exosomes. Exosome is all these cells, they have the ability that they are all the time. I have just yesterday I published a paper on, we call these exosomes as the garbage bags. They can use to consider that these exosomes, all these cells, they constantly keep throwing in the circulation some tiny little particles. We call them as nanovesicles. Now, their size is about 100 nanometers. Initially, they were considered as the garbage bags of the cells because they think any metabolite waste, metabolic waste, will be put into these small bags and thrown out of the cell. Now they consider that they are exosomes which are meant for intracellular communication. The one cell throwing these exosomes out in vitro, outside this cell, and it goes to another cell, fuses it there, and deliver the contents to the neuron. So what they are doing nowadays, they are making use of these exosomes. They culture the cells, take these exosomes which are thrown into the medium, and then load them with the help of the gene of interest and use this for transplantation or gene therapy. So this is the latest trends which are in the gene therapy vectors. Undesired effect, we have already talked about them. And thank you very much.
Thank you.
Any